Okay, nice. Okay. So today we will go over the data analysis part. And first is quality control, and then normalization, and selecting high variable genes, and then dimension reduction, as well as clustering, if we can go there. And so let's first recap what we have introduced yesterday. So the single cell RNA sequencing, this technique is starting from a tissue of interest or a sample. And then first we need to dissociate the cells and then isolate the cells using some technology such as microfluidic. And with this single cell, we then can break the cell walls and extract the RNA, mRNA sequencing. And that's, if you remember yesterday, that those mRNA is captured by the beads, which have the DNA oligo on the bead. And the DNA oligo has the sequencing for single cell barcode and molecular identifier. It's called UMI. And then with this, the next step is to, because this one, the mRNA is not stable, and so we need to reverse transcribe into cDNA, which is more stable, and also we can amplify them using PCR. And the reason we amplify this DNA sequencing is because any thought? Can you repeat the question? Why do we amplify the signal of the DNA?
Because each cell only has 1 picogram RNA.
Yeah, yeah. So because each cell only have 1 picogram or mRNA, and the capture rate is only 5 to 30%, so which is even less. Then to the detection threshold is usually 0.1 to 1 microgram for the sequencing core. So to reach that threshold, we need to amplify it. That's why we do the PCR. And after we send into the core, then we will get the sequencing files from them. And the sequencing files give us the barcode for each cell, the molecular UMI to identify which mRNA it comes from, as well as the sample ID, so which sample it comes from. And then using software such as Cell Ranger, we then can get the gene expression matrix, where each row is a gene and each column is a cell. And then we can do further data analysis. So today we're gonna focus on this part, that from the expression matrix, we will do further exploration. So today let's introduce some key concepts that's important. And the first is the UMI counts, and that's the raw gene expression counts based on UMI per gene and per cell. And that's the matrix entries. And so what does this mean biologically?
The UMI count means
Yeah,
the number of RNA that was sequenced.
yeah, the number of mRNA that's sequenced. Yes, great. And then the another concept is the cell library size, and that's the total number of mRNA molecules and detected per cell. So that's so for each column, that's a cell. So the taking sum of that cell, and that's called a cell library size. And another concept is the sequencing depth. So there is two type of sequencing depth in the single cell RNA sequencing analysis. One is per cell sequencing depth, and that is the cell library size. So basically the sum of each cell. Basically how many total counts of mRNA for that cell that's sequenced. And another is per gene sequencing depth. That is the reads. That is the sum of the gene. And for each row. And so these concepts are important as we will use these for data analysis and also for the normalization. And before we going further, so let's take a look at what's dropouts. So as we mentioned yesterday, that these we starting from a sample, and this sample is full of like different cells, and within each cell we have these DNA and which will initiate the transcription and transcribe into the mRNA, which may decay and then become stable or whatever we observed in the sample, and that's the biological process. And then when we sample, we actually, when we sequencing, we actually taking, we only observe this part. And then, so as we can see, this is like doing the cDNA and reverse transcription and then PCR amplification, and then sending to the sequencing core. So this is corresponding to different steps of RNA sequencing. So the dropouts means that we observed the zeros in the sequencing readout. And so, however, these observed zeros, they may have different reasons, as summarized here. For the first case is, for example, one gene that's not initiated transcription at all. Those may be silenced genes. For example, maybe in the immune cells, the developmental gene won't be expressed, or the differentiated cells, the division genes won't be expressed. So those genes that just silenced. So because there is no initiation, then there is no mRNA, and we won't be able to reverse transcribe into cDNA or sequence. So we get 0. And that 0 is a biological 0. It means true information. And so that's a 0 that it's a good, that we care. So it reveals some information. And the second case is that some genes, it initiated. However, it's either because like it's too fast decay, or some other reason that it's not stable. So we won't see any mRNA in that cell. And then the sample is 0, so we won't get any signal as well. So that's also a biological 0. So all of these two is true zeros that exist in the cell. And the third case is where the mRNA is initiated, transcribed, and also stable exist in the cell. However, it cannot reverse transcribe into DNA, as we mentioned that some sequencing have different efficiency on binding oligos, etc. And so then we because here the cDNA we do not capture, so there won't be any sequencing. So that's a technical zero. And the fourth case is that, okay, we have the stable mRNA in the cell, and we can reverse transcribe into cDNA. However, when we do PCR, because each mRNA have different sequencing, so they have different efficiency of PCR amplification, and some sequencing is very low efficiency, so we won't be able to amplify it to the detecting threshold for the sequencing core. So we still get 0. So, and that's called sampling 0. And these two case is so this two case is similar. So here is that it's not amplified at all. Here is it's amplified, but because the original signal is too weak, so even it's amplified efficiently, the total amount is still doesn't reach the threshold to be detected. So we will still see a 0. So this two called sampling 0. It means that there is true signal and there is a true cDNA, but we cannot we did not observe that. And the fourth one is what we see non-zeros. So it means that there are stable mRNA and successfully reverse transcribed and successfully amplified to be detectable. And then we see that one. And all of these are dropouts. And some are the silenced genes, and some is because technical or sample reason. Okay. And so this is a data analysis pipeline, and which we will go over one by one today. And so the first part is gonna be quality control and then normalization and selecting genes and PC principal principal component analysis to reduce the dimension and then calculate the distance for clustering. Okay, so we can start from I upload the code yesterday, and we can okay, and let's open the R. Okay. So here is the code. And then yes, this one. And first, how many of you successfully installed the RStudio? Okay, great. And yeah, so, and also this library is required for us to run this code. So we can run this chunk to load the library. And then, so this part is we loading this data that we can see here. You need to download the data on the shared Google Drive, and then locally you need to figure out where you saved that, and then you can input this path where you saved that data, and then you input that data path and give to this parameter. And the read 10x, this is a function that can read this sequence the gene expression output gene expression matrix for the 10x genomics output, and then it will create an object and then you can save that in this data, in this variable, and then you can run this chunk. And then here is some exploration of the data. And as you can see, so this is first is a matrix, and then you can display the head of this data. So here shows the genes and the gene names, and you can plot the first 30 cell, and then you can see some values here. And here is some example that these two genes, so as we mentioned that each row is a gene and each column is a cell. So here is the first cell expressed this gene with 4 count, and the dot means 0, so it doesn't express any MS4A1 gene. And so for the second cell, it doesn't express this gene, but have a high expression of the second gene, and the third one, etc. So all of these dots are zeros, and these are some are mRNA UMI counts, the mRNA molecule counts for the corresponding genes. And then, so we usually need to work on this CRET object because it have a lot of developed functions that we can easily do the data analysis for that object. And so this is creating this object, and we using the data which we just read from the file, the gene expression matrix file, and then we input to this function, and then the project name is PBMC, and then we can initialize this object, and then we can see what this object looks like, and that's this part. And you can run this chunk on yours as well. And here is some informations about this data set, this object. So you can see, for example, here is a cell, is a cell barcode, and yeah, you can explore it yourself. And here is the gene names, and that's the most important that we interested. Okay, and here is some quick way to see some features, some the data, like the gene list is here, and yeah. Okay. So maybe everyone can run this part on your computer, and Okay. Let's keep going. At the end of first session today, we will have more time for you to play with it, and we can continue for next part, and then yeah. So next, what we will do is the quality control. So as we already see that there are dropouts, and also the dropouts is it can also yes, those are some are just low quality cells, and another things that we need to consider about quality control is so we have an expectation of what true biology cells, how many genes do we expect and how many counts, etc. And if there are too few genes or too few counts, it either means the empty droplet, which means it doesn't capture anything, as we mentioned yesterday, because this although it's well controlled, but it still have some probability that capture nothing, and or low quality of cells. So those ones, as we shown in the dropout slides, there might be a lot of dropouts in that cell. So we don't want those cells. And on the other way, we also don't want the cells expressed too many. And those could be doublets or multiplets. And here, for example, if it captured 2 cells, then it may show or multiple cells, it may show like a different cell features, and we also don't want to include those. So we want to exclude those cells as well. And some technical terms is the feature count is the number of the genes, and the RNA count is the number of the UMS. And so another quality control metric we need to consider is the mitochondrial RNA. And this is because during this step, when we preparing from the tissue, we will dissociate the cells by resolving or by removing the cellular matrix, extracellular matrix, and then isolate the to single cells. And during that that step, we may not only break the extracellular matrix, but also break the cells. And the cell may be too harsh, or there may be dying cells. And in those dying cells, they release their they release the mitochondrial RNA to the cytoplasm. So then in the normally in the healthy cell, we won't see that. However, in the dying cell, we will see a lot of this mitochondrial mRNA. So then for quality control purpose, we don't want those dying cells to mess up our analysis. So we also want to exclude those. And so for quality control purpose, we need to do focus on 3 metrics. One is the RNA count, and also the feature count. So this is the total. This is the mRNA, and this is the gene that expressed, and also the mitochondrial content. And so what we do is that we first can identify and remove those outliers, and also we need to keep in mind that different sample may require different cutoffs. Yeah, and let's we can go to the code. Let me to here. Okay. And so we just created this project and object and saved in this variable. And next, here is also display some just display some features of this object. And so the next chunk is doing this, getting the mitochondria percentage. So here is calculating the percentage. And then what we want is that we want to extract the mitochondria mRNA. So those is usually with a pattern of MT. So if one gene has a name MT in it, and then that's usually the mitochondria genes. mRNAs, and then we can calculate the percentage, and then we saved in this object. And here is we display the histogram or the distribution of this percentage of mitochondria. And so we can see. So here is percentage. This is 5%, 10%. And we can see the most of the cells have less than 5% mitochondria mRNA, and which means that's a good quality cells, that not dying cells. And some cells have more than 10%, so those are probably the dying cells. Yes.
So how do they distinguish mitochondria from the target? Where is that MT label from?
So if you see here is the gene's name, and yeah, I didn't display here. But if you list all the genes, there are some genes that have MT in that gene's name, and those genes are usually the mitochondria mRNA. Yeah, and so here is just saying that if a gene have that pattern, and then you calculate the percentage. Okay, and so now we have this mitochondrial distribution. And next, what we want to do is that we want to see the distribution for mitochondria mRNA, and also the number of feature, and as well as the number, the count, the total count. And so the total count, just a reminder, that's the total mRNA molecules. The number of feature is the non-zero, the expressed genes total number. And then we can plot those. And then we here is that we also can display the scatter plot to see how two features, for example, the count of mRNA versus the feature like the genes expressed, what's their relationship, as well as we can see the number of counts versus the mitochondrial percentage. And yeah, let's skip that one for now, and we can run this chunk. Okay, now we have this one, and first is the violin plots of these three different features, and you can see the distribution of the total amount of mRNA, and so it's on average it's 2000-ish or 2500 expression, and most of them is less than 5000, and some outliers goes to 15,000. And those, what would you think those cells could be? Like, yeah, doublet or multiplet. Yeah. So those outliers is probably the cells that express the two different cell types or etc. that the bead or the droplet captured like two cells or more. So we want to exclude those data points. And here is how many genes are expressed for each cell. And we can see on average approximately 800-ish genes are expressed for the majority. And most of them are less than 2,000 genes expressed. Still some outliers have more than 3,000 genes expressed. And
I'm sorry, could you say again what the difference between end count and end feature is?
Yeah, so this the count means the UMI count.
Yeah. Oh,
Yeah, so that's the total mRNA. And the feature means that the non-zero genes. Yeah.
and
So
each dot is one cell.
each dot is one cell. Yeah, each dot is one cell. And the third one is the percentage of the mitochondria mRNA, and still each dot is a cell. And we can see that on average that 2% or 3% mitochondria we can observe from each cell, single cell. And most of them are less than 5%. Any questions here? Okay. And so next thing is here is that we calculate some statistics. So how do we define those outlier? We need to find a cutoff or a threshold. And one way is that we can find figure out the quantile. And here we can calculate the 97% quantile and for each metric. And then those cells which are higher than this quantile, we consider them as the outliers and or too much expression. And then we want to exclude those cells. So if you run this one, and if you run this one, you can see the result. Here. Yeah, so the 97%, so for example, for the mRNA count and the 97 quantile is this number, 4,800. So that means that with this threshold, that's a 97% quantile, and all of these are higher than that value. And also as for the genes, for the feature number, we can also get that value, and that's almost 1,500. And so which means that the like 1,500 is here, and all of the cells above these are higher than 97%. And also we can get the for the mitochondria mRNA, that's 4.6%. So here the value is approximately here, and those cells will be considered as higher. And so And the next one we can explore is this scatter plot, and we can plot these features together. For example, here in this one, we're plotting the UMI counts and the feature counts for each cell. And so the x-axis is the UMI counts, and the y-axis is how many genes expressed in that cell. And we can see each dot is a single cell. And we can see that this is it's almost a line, which means that they are positive correlated, and the coefficient is 0.95. And that makes sense because we can imagine that the more the more genes expressed, the more UMI counts we would expect to observe. And we can also scatter plot the UMI counts versus mitochondrial mRNA. And here is almost no correlation. If there is, then it's a negative, very weak negative correlation. And so also still each dot is a single cell, and the x-axis is the UMI counts, and the y-axis is the mitochondrial percentage. And there are weak correlation. We can also expect that the healthy cells probably have less mitochondria, and those cells are dying. Then because the cell dying, then the cell cannot maintain a good environment for mRNA to be stable. So there is less. So that's also makes sense, but it's almost no correlation. And so that's, we explore the data a little. And so for, yeah, and for the, so I just want to explain this a little. So this is the data. And as we mentioned here, this is different genes. And then each column is different cells. And we can see a lot zeros. All of these are zeros. And you occasionally see some ones. And yeah, it's all of these. So it's a very, very sparse matrix. You can imagine the original data is yeah. And so after we got that, and then we can further explore this one. So here is that we calculate how many cells before we do the filtering. So there are in total 2,700 cells. And then here is we do the quality control, which means we remove those outliers, remove those low quality cells. And what it is doing is that we have some threshold decided by the quantiles, which is number here. And we use that number as a threshold. And we only select the UMI counts is less than 97%, and the feature count is less than 1499. And also, we don't want like dropouts. We don't like those cells have very low expression, or maybe they are dying, or they are just not healthy cells, etc. So we also want they have minimal expression, and usually we use the genes 200, and this depends on your purpose. And by default, we can use 200 genes at least expressed in that in all the cells that we will analyze. And also we want the mitochondrial mRNA, that percent is also less than the threshold defined by the 97 quantile. And so that's 4.6. And this one will exclude all the outliers and only preserve these past this filter. And then we use them for downstream analysis, and that's quality control. And so this is before we filter them out. This is, and then we can calculate after we filter out those low quality cells, how many cells we have left. And we can see here, it's removed 180, 70 cells. And so that's a quality control. And so to summarize, that metrics, the three metrics we use for quality control is MRA count or UMI count, and then the feature count or the gene count, and also we use the mitochondrial content percentage. And we need to identify the outlier. So we need to define a threshold. And one way is we can use a quantile, like 97, 95, depends on your purpose and also depends on how the data distributed. And also different samples may require different cutoffs because especially their lab preparation are differently, and so they may have different qualities, etc. And sometimes there are, if a very rigorous experimental design, they will have spec-in. And what are the spec-ins? It's basically when they do this sample preparation, and they will have some mRNA. That mRNA is already, they know the concentration or the amount of the mRNA, and then they can mix with the sample and then do the further lab preparation step until they got to the sequencing and the gene expression metrics. And because they already know those control, the spec-in mRNA, those concentration or this amount, and so they can see from these gene expression metrics and see whether what they measured aligns with the original amount. And if it aligns, then it means a good experimental pipeline, and then the data is more reliable. And so they sometimes also use these spec-ins for quality control purpose. And if you plot this spec-in mRNA, like the original concentration or amount of at the axis, and the y-axis is what after sequencing and what you get from the data analysis, and it should looks like a line. So they should be highly correlated, and then that's a good like a sample. And okay, so let's take a break and we come back at 10, and then we will start in doing those code yourself for a little, and then I will walk around to make sure that everyone catch this part, and then we will move on afterwards. Okay. So there is an attendance sheet, a sign sheet. If you haven't signed, please sign your name.
Yeah, I wanted to ask, when you were talking about the dropout slide. Wouldn't it still capture like a lot of that noise? And then if you have like signals that are just like very, or if you have like RNAs that already have very low signal, wouldn't sequencing deeply not help? If RNA splicing is noisy?